This timeline tracks the historical pace of vaccine development. For diseases like polio or rabies, isolating the pathogen, testing, and scaling manufacturing took decades. Yet, in the year 2020, the global scientific community engineered, tested, and distributed effective immunizations in under 11 months. To achieve that speed, scientists had to identify exactly what the immune system needed to see. The biological objective of any vaccine is to safely present a specific foreign antigen to the body to trigger a lasting defense without causing the disease itself. For SARS-CoV-2, this model shows the target epitope required for immune recognition, the spike protein. It's the distinct structure the virus uses to breach human cells. If the immune system learns to recognize this specific spike, it can neutralize the actual virus upon infection. Teaching billions of immune systems to recognize this structure simultaneously required manufacturing at a global scale. Relying on a single method would guarantee supply bottlenecks. Instead, researchers utilized a diverse portfolio of technologies. If we look closely at the vaccines distributed globally, they divide into two distinct engineering solutions. First is endogenous synthesis. Instead of injecting the protein, it delivers genetic instructions, turning the host cells into bio-reactors to build the antigen. Second is exogenous delivery. This relies on external laboratories to manufacture physical pathogenic payloads, which are then injected directly into the body. Understanding how these distinct cellular pathways operate explains how global health authorities successfully mapped an array of diverse technologies against a unified biological threat. The first category of endogenous synthesis utilizes messenger RNA, or mRNA. While studied for years, the pandemic marked the first time this specific technology was authorized for widespread use in humans, moving from clinical trials to global distribution. This platform was driven globally by two major formulations. The first was developed jointly by Pfizer-BioNTech. The second was developed by Moderna, with both brands seeing distribution across the US, Europe, and the rest of the world. The central engineering challenge with this platform is chemical stability. Bare mRNA is fragile. If injected directly into the human bloodstream, enzymes would degrade the genetic code before it ever reached a cell. To solve this, scientists encapsulate the mRNA inside a lipid nanoparticle. This specialized fat bubble ferries the genetic instructions through the body, protects them from degradation, and fuses with the host cell to deposit its contents. Looking at this cellular cross-section, you can see that once inside, the mRNA strand is released directly into the cytoplasm. This is the fluid area that contains the manufacturing machinery. Crucially, the genetic material of the host resides entirely within the nucleus. The mRNA sequence is blocked by the nuclear membrane, meaning it has no capacity to alter the host's DNA. Instead, the cell's natural ribosomes intercept the mRNA. They begin translating this transient instruction manual, using it to synthesize the harmless SARS-CoV-2 spike protein. Once manufactured, the cell anchors these newly synthesized spike proteins to the outside of its own membrane, displaying them to the surrounding environment. The body's patrol units immediately recognize these spikes as foreign. This triggers a humoral immune response, generating targeted antibodies, alongside a cellular response, activating T cells for memory. By co-opting the host's ribosomes to do the heavy lifting of protein manufacturing, mRNA vaccines bypass external lab synthesis. After a few days, the cell naturally degrades and disposes of the synthetic code. The second method of delivering genetic instructions is the viral vector platform, which gained a global footprint through the Oxford-AstraZeneca vaccine. This approach was also engineered into a single-dose option by Johnson & Johnson. Instead of using synthetic lipid bubbles, this platform uses a biological Trojan horse known as a recombinant adenovirus. An adenovirus is a common pathogen that normally causes mild human cold symptoms. First, researchers take this adenovirus and genetically modify it so that it cannot replicate or cause illness in humans. It is effectively hollowed out. The second step involves inserting the DNA sequence that codes for the SARS-CoV-2 spike protein directly into this viral shell. When injected, the adenovirus acts as a delivery vehicle. It latches onto host cells, breaches the membrane, and unloads the genetic payload. This mechanism of utilizing modified human adenoviruses was also the foundational architecture for the Sputnik V vaccine developed by the Gamaleya Institute. Once the vector unloads its sequence, the process mirrors the mRNA pathway. The host cell reads the code, builds the spike proteins, and displays them on its surface to train the immune system. Both mRNA and viral vectors rely on this genetic recombinant approach. By delivering instructions instead of physical proteins, they bypass the logistical hurdles and biological risks of growing quantities of live pathogens in a laboratory. The second major immunological approach relies on exogenous delivery. These are traditional methods that deliver pre-made components of the virus directly into the body. Within this category is the protein subunit category, championed during the pandemic by Novavax, which secured approvals across the US, Europe, and India. Subunit vaccines contain no genetic instructions and no whole viruses. They contain only the isolated spike proteins themselves, manufactured in external bioreactors. Because this method delivers an isolated protein, it is exceptionally safe. However, this purity creates a hurdle. On their own, these solitary proteins often fail to agitate the immune system enough to trigger a lasting memory response. To solve this, manufacturers introduce an adjuvant. This is a secondary ingredient mixed directly into the vaccine. Its purpose is to locally stimulate the immune system at the injection site, ensuring that the body's defenses swarm the area and process the purified proteins. This combination of isolated proteins and immune stimulants was the basis for other global vaccines, like Corbevax, manufactured in India. While subunit vaccines provide stable safety profiles, they trade the speed of genetic instruction for the complex reality of scaling up physical protein manufacturing. The final category represents the most historically proven method in immunology: inactivated whole virus vaccines. This architecture was a tool for global players like Sinovac, which saw deployment across Asia, South America, and Africa. Unlike the targeted genetic or subunit approaches, this method requires industrial scale. Actual SARS-CoV-2 viruses must be grown in vast, secure biological laboratories. Once cultivated, the viral yield is exposed to heat, radiation, or chemicals like beta-propiolactone. This process kills the virus, destroying its genetic core while leaving its physical exterior intact. Because the pathogen is dead, it is safe to inject. It has no biological capacity to replicate or cause COVID-19 in the host. This approach provides an immunological advantage because the immune system is presented with the entire dead structure. It mounts a defense against multiple parts of the virus's anatomy rather than focusing exclusively on the spike protein. This whole-agent methodology also powered Corbevax, providing a stable immunization option throughout India. While these inactivated options often require booster doses to maintain antibody levels, their stability and historical safety record provided a backbone for the global distribution effort. This matrix shows the 4 distinct architectures that mobilized against the pandemic. On the top, the endogenous synthesis of RNA and viral vectors. On the bottom, the exogenous physical delivery of subunits and inactivated virions. The biological mechanisms of these platforms dictated their real-world logistical constraints. How the payload was built determined how it had to be shipped. The fragile lipid nanoparticles protecting RNA code required ultra-cold freezers, initially limiting their reach in developing regions. Conversely, the stable proteins and dead virions of traditional methods could be transported in standard commercial refrigerators. For countries with existing vaccine supply chains, the traditional inactivated and subunit methods integrated into the infrastructure, offering a predictable defense. Meanwhile, when time was the single most critical factor, the next-generation technologies proved their value through speed. By delivering instructions instead of physical payloads, mRNA and vector platforms moved from genomic sequencing to clinical trials in a matter of weeks. Ultimately, global health security depends on matching technology to the logistical environment. Surviving a pandemic requires a scientifically diverse portfolio, utilizing both the rapid delivery of instructions and the stable delivery of physical payloads.